All right, so this video is on Duchenne Muscular Dystrophy. As the name suggests, it does involve a wasting or degenerative process of the muscles, especially the skeletal and cardiac muscles. It can be quite devastating. It's debilitating. It starts at an early age. Of the dystrophies that involve the protein dystrophin, it's pretty much the worst one. I'll get into that a little bit more, but let's just jump into it in terms of the first presentation of the disorder is typically at age 3 to 5 years. There's weakness in the hips and trunk, unusual or waddling gait, frequent falls and difficulty rising and using upper limbs to get up in circumstances where normally for most kids that age that wouldn't be needed, enlarged calf muscles, and just to note that it is an X-linked recessive disorder, so it's going to affect mostly folks who are genetically male, XY, and rarely, though, folks who are developmentally female and have an X0 karyotype have also been known to present with Duchenne muscular dystrophy as well. So in terms of progression without treatment, the boy may require a wheelchair by age 11 to 12. Treatment with steroids like prednisone helps maintain muscle strength and function for a little bit longer. Swallowing dysfunction, chronic respiratory insufficiency, which can be quite devastating, and dilated cardiomyopathy is present pretty much universally by age 18. And life expectancy is increasing, though most still pass away, sadly, in their 20s. So why does all of this happen? Well, as we covered in class, dystrophin is a key protein linking the actin cytoskeleton of muscle cells, muscle fibers, to laminin proteins of the extracellular matrix, so the basal lamina of those that surrounding connective tissue, like in the endomysium. And simplified here, of course, there's lots of other proteins around, but there's this sort of chunk here, sometimes called the dystrophin bolt, sort of like a rivet that really ties the actin cytoskeleton of the muscle fiber to the basal lamina here, to the laminin protein in the basal lamina outside the cell. And that's made up of alpha dystroglycan, beta dystroglycan, and dystrophin. And dystrophin, being a very large protein and having a very large and complicated gene, is susceptible to mutations. And that's why this dystrophin is so often involved in these kind of disorders. In Duchenne's, this is pretty much completely knocked out. So, which you can see here in this fluorescence immunohistochemistry, where here in the normal control, the dystrophin is localized to the sarcolemma. This is a cross-section. This would be, tangent to the endomysium here. And in the person with Duchenne's, it's just not there. It's gone. So the exact mechanism of the degenerative process here, the atrophy, is unknown, but it's believed to originate from, the mechanical stress, maybe something about the uncoupling of the connective tissue and the muscle fiber with the loss of that dystrophin bolt, as it's called. So getting into the histopathological features now, I just have an image here from a histology guide that we should all be familiar with, and then the second one there. So I'll just go down, and if we look here, the paramysium is pretty, it's there, it's visible. Endomysium, this is on the healthy one. We know it's there. It's not super prominent. There's like a good sense of structure. You can kind of see the cells have a good shape to them. Whereas here in Duchenne's, this looks quite severe, quite progressed. Just looks very chaotic. There's a lot of this. This might be paramysium here. The connective tissue, paramysium, endomysium here is it's proliferated quite a bit. It's a lot of fibrosis, which means really decreased muscle volume, ultimately decreased tissue that's capable of producing force, which is where you get that weakness that is quite characteristic. You can see how much less muscle is actually there. You also can see a diffuse infiltration of adipocytes here into the tissue, and unusual variation in fiber size. You can see very small ones and very large, which is not what you see here. Some of these might be type 1 or type 2, but here, small versus large is not dependent on what type of fiber at all. The fiber borders tend to be more rounded instead of polygonal, so less structure for sure. And there's a greater number of nuclei compared to the healthy controls that are visible. But also you'll notice these areas where there's a whole bunch of nuclei bundled together, and this is actually a necrotic fiber. Those are lymphocytes or phagocytic cells that have been infiltrated that fiber itself. And so those are visible in a few different places with a pale cytoplasm. These might be regenerative fibers here. Regenerative fibers are basically immature cells that are sort of, they're growing back. Then we also have these hyalinized which means they're sort of glass-like. They don't have a texture. They've lost their striations. That sort of sarcomeric structure is not very clear. And they're also hyper-eosinophilic, so that's a pretty recognizable feature in Duchenne's, these hyalinized fibers. I hope that was interesting and informative. Here are my works cited, and thank you so much.